Okay, welcome back. In the last video, we looked at the structure of neurons, cells, and synapse. Now we're going to talk about neural communication. We said that the way information is transmitted in the nervous system, it's called the electrochemical messaging system. There are two ways information is transmitted in the nervous system. One is through electrical signals. The other one is through chemical signals. Information in a given neuron is transmitted through electric signals, and in between two neurons through synapse, it is through chemical messaging. First, we'll talk about electrical messaging system. Then we'll talk about the chemical messaging system. The electrical messages generated in the neuron are called neural impulses or action potentials. The neuron is at a resting phase when not carrying information. It has a negative charge inside and a positive charge outside. In fact, there is a potential difference between the inside and outside of the axon, so much so that there is always a potential difference of negative 70 millivolts. And this is called the resting membrane potential. Now, think of battery, and when you look at it, let's look at this one. This is a battery, and there is a positive charge on one side and a negative charge on the other side. And if you look at it carefully, it says 1.5 volts. That means there is a potential difference in charge between these 2 points, and the difference is measured in volts. It's 1.5 volts. Now we're talking about neuron here, so potential difference is not that big. It is in millivolts. A millivolt is a thousandth of a volt. So the inside of the neuron is negative and the outside is positive, and the potential difference between these two is negative 70 millivolts. And the reason why there is a negative 70 millivolts is because of the differential distribution of ions inside and outside the cell. Cells are embedded within a fluid. We call that extracellular fluid. So we have extracellular fluid outside the cell and intracellular fluid inside the cell. And there are ions inside and outside the cell, like sodium ions, potassium ions, proteins, and the way these are distributed across the cell membrane makes the inside of the cell more negative than outside, so that the potential difference at resting phase when the neuron is not carrying information is negative 70 millivolts. Now, let's look at a video that gives us information about why there is a negative charge inside and a positive charge outside.
During the resting potential, sodium ions, which carry a positive charge and are represented here as yellow spheres, are found mostly on the outside of the axon. Potassium ions, which also carry a positive charge and are shown here in green, are located mostly on the inside of the neuron. Large negative charged protein molecules are always found inside the cell. Other ions are present, but we will focus on the behavior of these three molecules. There are more negative charged protein molecules inside of the cell than positive charged potassium ions. The inside of the cell, therefore, is more negative than the outside during the resting potential. This changes with the arrival of the action potential.
So there are more sodium ions outside of the neuron and more potassium ions inside. And that is important to remember. What is action potential then? Well, action potential is this sudden reversal of the membrane potential on a specific point on the axon. Usually, action potential starts at this junction between the cell body and the axon called axonal hillock. That's where the action potential generates and it's propagated throughout the length of the axon. Action potential basically is sudden reversal of the charge on the membrane. Remember, it's negative inside and positive outside. But once a neuron gets stimulated, there is a sudden reversal of the charge. So the inside gets positive and the outside becomes negative. Um, you can see the resting membrane potential goes to being positive, which is positive 30 to positive 35 millivolts. This sudden reversal at this point is called action potential. And this is what is propagated from one point to another point of the axon, which is usually from one node of Ranvier to another node of Ranvier. And that's how the information gets transmitted in the form of sudden reversals of potential. So what is action potential then? It is a sudden reversal of axonal membrane's potential from negative 70 millivolts to positive 30 or 35 millivolts. The question is, how is this action potential generated and how is it transmitted through the axon? There are 4 or 5 stages of The first one is the resting membrane potential when the neuron is not stimulated. The second one is depolarization. The third one is the propagation. The fourth one is the depolarization. And the last one is the sodium potassium pump. We already know what resting membrane potential is. Just to remind you, it's the excess of sodium outside and the excess of potassium inside, with the membrane being negative 70 millivolts and the extracellular fluid. Now, first we'll talk about depolarization, then we'll talk about propagation, then we'll go back to these last two steps. Depolarization is this reversal of negative 70 millivolts to negative positive 30 or 35 millivolts, right? The question is, how or why does it happen? Well, when a neuron is stimulated, especially at this axonal hillock, right, there is a sudden reversal of the charges. The question is, why is there a reversal of charge at this particular point? If you look at this one, this is the particular spot. The sodium channels are gates for the sodium ions to go through. Remember, the cell membrane is semi-permeable. It only allows some, but not all things. Sodium ion requires special gates for it to enter the neuron. So once a neuron is stimulated, the sodium gates at this particular point, they open, and there is a sodium influx. In other words, sodium moves into the neuron. And what happens when inside is negative and you're adding positive sodium ions, the negative charge gradually decreases. It goes from negative 70 to now 60, negative 65, 60, and so forth. Once it reaches negative 55 millivolts, in other words, if the stimulation is strong enough that it leaves the sodium open for it to reach this potential difference, then more sodium ions open, and there is this influx of more sodium ion, where the negative 55 becomes negative 50, 45, 30, 0, then positive. It eventually goes to positive 30 or 35 millivolts. That is called action potential. So there is action potential generated at one particular point, or the axonal hillock on the axon. Then what happens is, this particular um, action potential is transmitted along the length of the neuron from one node of Ranvier to another node of Ranvier. This is called propagation. So the action potential is generated here. Now, through a process called induction, it induces a similar change in the next node of Ranvier, which again induces the change in the next node. And this is how the reversal of the charge continues along the length of the neuron. This is called propagation of neural impulse or action potential. In other words, it is how the information is transmitted along the length of the axon. Now, what happens is the third step, which is repolarization. A quick video about how the information gets transmitted along the neuron once it is generated.
The action potential originates from the axon hillock on the cell body. Once initiated, the action potential travels unchanged all the way from the axon hillock to the axon terminals. Events responsible for this all or none propagation of the action potential occur along its membrane. Here, in a crisscross section of the axon membrane, we see the movement of sodium ions, shown as yellow spheres, into the cells as the action potential travels down the axon. As the action potential moves along the axon, brief changes in voltage across the membrane cause sodium channels, which are embedded in the axon, to open. Positive charged sodium ions move from the outside to the inside of the cell. The positive sodium ions move inside the cell because they are attracted by its negative charge. After the action potential passes, the sodium channels close.
Now, what happens to the positive charge here once it is transmitted to the next part? Well, it goes through the next step, and it's called repolarization. Repolarization is when this positively charged membrane, this positive 35 millivolts, changes to negative 70 millivolts back to its original resting membrane potential. The question is, how does it happen? Well, it happens through um, potassium efflux. In other words, the potassium ions that are more in the neuron, they come out of the membrane or the axon. So what happens when the positive ions leave the neuron becomes more negative. From positive 35, it drops to negative 70 millivolts. So the question is, what is the reason for depolarization? In other words, the resting membrane potential changing to positive 35, it's influx of sodium. What is the reason for repolarization, which is the resting membrane potential with the positive 35 millivolts goes back to the negative 70 millivolts? It's efflux moving out of the potassium. It's potassium moving out of the cell membrane or the neuron. Now, the action potential generation follows all or none law, which means that when a neuron is stimulated, whether there is an action potential that is generated, if it is, then it is propagated all along the length of the neuron, or it is not generated, which depends on the strength of the stimulus. Remember earlier we said that if the sodium ions open long enough for the resting membrane potential to reach negative 55 millivolts, then that's a threshold level. Once it reaches that level, then it automatically action potential is generated. In other words, more sodium ion will enter the neuron and it reaches positive 35 millivolts. The initial stimulation doesn't result in difference in potential of negative 55 millivolts, then there is no action potential generated. That's why it's called all or none law. Whether it is generated or not generated, there is nothing between half generation or three-fourths of action potential. It all depends on whether or not the stimulation is sufficient enough for the ions to influx into the neuron to an extent that it reaches a threshold of negative 55 millivolts. So we looked at depolarization, propagation, and at a given point, again, repolarization, where the negative charge is restored. Now, the question is a neuron ready to fire again, given that it is negative 70 millivolts? Do you think the neuron would be ready to fire again? Just think of this. This is the neuron, this is stimulus, action potential that is generated here, right? And it is propagated to the next node of Renvier. And that when that happens, this particular first part actually reverses its polarization by going through repolarization through potassium efflux. Remember that? And the next point, it goes to the next point, it continues. It's like if you think of, I usually students stand say, okay, this is negative 70 millivolts. Okay? Resting membrane potential. This is positive action potential. Like clap is an action potential. So I make students stand in a line and say, you're the first node of Renvier, you clap, right? The next node of Renvier is unclapped. So there is a wave of clapping, it's like ping, ting, ping, ping, right? And when there is this wave of clapping, so the first one claps, the second one also claps. The moment the second one claps, the first one goes to repolarization. So it goes to negative 70 millivolts. So it's like a wave of windows clapping and unclapping, or the windows closing and opening. So the point is this: now that all the parts on the neuron are at a stage where there is negative 70 millivolts back to resting membrane potential, here's a question: is this neuron ready to fire again? Yes, I'm asking you. Just think about it. You're right. No. Why? Why is it not ready? Well, for it to fire again, it should be able to move the sodium ions outside. Remember, the depolarization is due to influx of sodium, but the sodium is trapped inside the neuron. So there is this next step called sodium-potassium pump, where the excess of sodium inside is pumped out and the excess of potassium outside is pumped in, so that it's not only 70 millivolts now, but it has appropriate amount of sodium outside and appropriate amount of potassium inside for it to go through the process of depolarization, repolarization again. So is the neuron ready to pump again? No. This is because of ionic imbalance. Well, that was electrical messaging. In the next video, we'll talk about chemical messaging system. Electrical messaging is how the information is transmitted along a given neuron, whereas chemical messaging is how the information is transmitted between any two given neurons through this gap called synapse.